Good morning, students. In the previous class, we had studied about the human excretory system. We had seen that the chief organ of excretion are a pair of kidneys. We had studied the longitudinal section of the kidney, showing us that nephrons are the structural and functional units of kidney, and each kidney has about 1.2 million nephrons, also called urinary ferrous tubules. Then we had seen in the longitudinal section of the kidney the placement of these nephrons in the renal cortex and the renal medulla. Today we will go into the detail of the structure of a nephron. As I have told you, each kidney consists of a large number of nephrons. Almost about 1.2 million nephrons are present in each kidney. This nephron is also called a urinary ferrous tubule, and it is the structural and functional unit of kidney. Each nephron consists of 5 main regions, and it is an independent unit and produces a very small amount of urine. And this urine is collected by various ducts from all the nephrons and then finally poured in the renal pelvis. So the 5 regions of the nephron are the renal corpuscle, also called the Malpighian body, which is composed up of the cup-shaped Bowman's capsule, and in the hollow of the cup there is a network of capillaries called glomerulus. Together, Bowman's capsule and glomerulus make up the Malpighian body or the renal corpuscle. This leads into the proximal convoluted tubule, which is abbreviated as PCT. I will be telling you details about each part with reference to this diagram shown to you here. But first, I am just telling you the 5 major parts of the nephron. The PCT region of the nephron leads into the loop of Henle. Loop of Henle consists of the descending limb and the ascending limb, which then opens into the distal convoluted tubule, abbreviated as DCT, and finally DCT opens in the collecting tubule. This figure here shows you all the 5 regions very clearly. You can see the Malpighian body composed up of the Bowman's capsule and the glomerulus. Then it leads into a convoluted part of the tubule called the proximal convoluted tubule, which then leads into the loop of Henle, which has the descending limb and the ascending limb, and then the distal convoluted tubule opens into the collecting tubule. These 5 regions are clearly marked in the diagram. Now we will be discussing each of these regions one by one. To begin with, renal corpuscle or Malpighian body, as I told you, is composed up of the Bowman's capsule. Bowman's capsule is a cup-shaped hollow structure which is lined internally by 2 epithelial layers, and in the hollow of the cup it encloses a network of capillaries which is called the glomerulus. Now the glomerulus is a network of capillaries. It receives blood from the renal artery, as you can see in the diagram, from the renal artery through a branch which is called afferent arteriole. Please make note of the spellings here. Afferent is spelled with A and afferent arteriole then breaks up to form a capillary network, and the blood leaves the glomerulus through the efferent arteriole. Why I said make note of the spellings? Because the blood vessel that leaves the glomerulus is called efferent arteriole, whereas branch of renal artery which forms the glomerulus is afferent arteriole. The efferent arteriole is narrower than the afferent arteriole, and the efferent arteriole, as you can see in this figure, then breaks up into a network of capillaries which surrounds the entire nephron. Whole of the PCT region, the loop of Henle, the DCT region is all surrounded by the efferent arteriole, and finally it joins to form the venule, which then opens into the renal blood. So renal vein. So the blood is returned into the renal vein. The renal artery brings blood, and the blood is sent back into the renal vein. You can see it in the figure how renal artery breaks up into afferent arteriole to form the glomerulus, and how efferent arteriole leaves the glomerulus, forms a capillary network on the tubule, and from there it joins to form the venule, which then returns blood to the renal vein. Then coming to the PCT region, that is the proximal convoluted tubule. This is the first coiled tubule that leads from the renal corpuscle, and it is twisted around itself, present in the cortex region. It is lined with a cuboidal epithelium, and the cells have numerous microvilli to increase the surface area. The mitochondria are concentrated near the basal surface that allow reabsorption of salts by active absorption. It transports the glomerular filtrate from Bowman's capsule into the loop of Henle. Now, you saw the figure. This is a detailed figure of the glomerulus, which then leads into the proximal convoluted tubule. You can see the cavity of the glomerulus lined by two layers of epithelial tissue. The loop of Henle is a U-shaped loop which continues from the proximal convoluted tubule, and it descends down from the cortex into the medulla region, which I had shown you on the last turn in the diagram of longitudinal section of the kidney, that how the loop of Henle descends down from the cortex into the medulla region, and again it returns back, forming a loop. The descending limb of loop of Henle is relatively thin, it is long, and it is freely permeable to water. It is lined with flattened cuboidal epithelial cells with very few microvilli and mitochondria, whereas the ascending limb of loop of Henle is comparatively thicker. It is composed up of flattened cuboidal epithelium with sparse microvilli and mitochondria, and it is impermeable to water and sodium chloride. Then the loop of Henle leads into the DCT region of the nephron, that is the distal convoluted tubule, which is the second coiled and twisted part of the tubule that continues from the ascending limb of loop of Henle and is then located in the cortex region of the kidney, in the renal cortex. It is relatively shorter in length and it is lined by the cuboidal epithelium. This leads into the collecting tubule, which is a sort of a part of DCT region only. The collecting tubule continues into a short straight the DCT continues into this short straight collecting tubule, which then several collecting tubules then open into a large collecting duct which passes downwards from the cortex to the medulla region. It is lined by cuboidal epithelial cells. Now, these collecting ducts, they join to form the larger ducts of Bellini in the medulla region of the kidney and run through the pyramids, and they ultimately open in the renal pelvis, draining the urine collected from different nephrons. The network of capillaries which was formed from the efferent arteriole is called vasa rectae, and these are parallel, wide, thin-walled and straight capillaries which arise from the efferent arteriole. They run parallel to the loop of Henle and the collecting ducts in medulla, and from medulla they loop back into the cortex region and they finally drain their blood into the venule. Vasa rectae help in retaining reabsorbed ions and urea by the medullary interstitial fluid, thereby maintaining high osmotic pressure. And their parallel arrangement helps in the concentration of urine in the loop of Henle. So this was the description of the nephron which I had shown you here in the diagram, the 5 regions of the nephron, that is the renal corpuscle or the Malpighian body opens into the distal convoluted tubule, which then opens into the loop of Henle having a descending limb and an ascending limb. The ascending limb opens into the distal convoluted tubule, which then opens into a short collecting tubule, and as you can see in this figure, the collecting tubules of several nephrons join to form the collecting duct, and this collecting duct from the renal cortex passes into the medullary region and opens and drains the urine in the renal pelvis. This figure shows you everything, even the duct of Bellini, which is formed after the collecting ducts have joined together before opening into the renal pelvis. Now, based on the position of these nephrons, there are 2 kinds of nephrons. These are the juxtamedullary nephrons and the cortical nephrons. Now, this is the figure which shows you the arrangement of these. Juxtamedullary nephrons. These nephrons, as you can see in this figure, one cortical nephron and one juxtamedullary nephron is shown to you, showing you portion of the kidney in the longitudinal section, showing the cortex region and the medullary region. So you can see the difference between the two, which I have tabulated here. You can see that the juxtamedullary nephron has long loops of Henle, whereas the cortical nephron has a short loop of Henle. Then in juxtamedullary nephron, these loop of Henle are placed deep down in the medullary region of the kidney, whereas in the cortical nephron they run to a very short distance in the medullary region. The glomeruli of the juxtamedullary nephrons are placed close to the inner margin of the cortex, whereas they are in the cortical nephrons, they are placed in the outer margin, in the outer cortex. The juxtamedullary nephrons are lesser in number and they constitute only up to 15% of the nephrons in the kidney, whereas the cortical nephrons are several and they constitute about 85% of the nephrons in the kidney. The majority of nephrons are the cortical nephrons. And the characteristics we have discussed, the placement is shown in this figure, how the cortical nephrons are in the cortical region and juxtamedullary nephrons have long loops of Henle and extend into the medullary region. So today we have studied about the structure of a nephron in detail with all the 5 parts and their characteristics and the kinds of nephrons and placement of nephrons in the kidney. This much is enough for today. On the next turn, we will be discussing the processes involved in the formation of urine. So I'll end today's class here. Take care and stay safe and healthy.